Thank you. Good afternoon. My name is Brato. Most of you probably don't know me. I've been here for 1 month or so. I'm possibly the newest member in the family. I work at the interface of fluid dynamics, soft matter, and biophysics. So today I will tell you about a problem of intracellular flows that involves some flavors of all these three subjects. So humans have roughly 10 trillion cells and 200 different cell types. Yet there is very little variation in the cell sizes, with majority of the cells being roughly in the range of 2 to 30 microns. Now, what is quite interesting is that this size variation is also true in a mouse and in an elephant. So you may suggest that there is some sort of evolutionary pressure on being the right size. And this can be rationalized by realizing that for cells to function, you need to transport or move things around. And for that, it relies on this inherently slow process of diffusion. And perhaps this diffusive timescale sets a constraint on how large a cell can grow. However, there are exceptions. X cells or oocytes are one of the largest known cell types in the animal kingdom, and this can be as large as some 500 microns to orders of millimeters. And in these very large cells, clearly you need a different mechanism of transport or mixing. And one way nature solves this problem is through cytoplasmic streaming. So streaming flows are actually not new. It was discovered around 250 years back by the Italian scientist Bonaventura Corti when he was studying algal plant cells called Cara corallina. In these algal plant cells, which you see over here, these are centimeters long. And in these tubular cells, you observe that there is a persistent circulatory motion of the fluid, which is drawn over here, all day, every day. And this circulatory motion of the cytoplasm is known as streamings. So as we know today, the streaming flows are rather generic in many other large cells, and examples include ciliates, algal cells, and slime molds. For today's talk, I'm going to tell you about streaming flows in the egg cells of the fruit fly Drosophila. For those of you who are not into biophysics, Drosophila or the fruit fly is often a model biological organism. And here I'm looking at its egg cell, which once fertilized will become the future embryo. So the egg cell develops over a span of 80 hours, and you see some snapshots of its developmental stages. The focus is over here when it's towards the end of its developmental stage. This black void that you're seeing, that's the egg cell. It's very large, it's 500 to 600 microns. And there is a large-scale vortical flow that emerges inside the cell that spans the entire cell chamber. You can probe this flow over here, as you can see from the structure, and you can be quantitative about it doing particle image velocimetry. What I really want to focus on, how this streaming flow emerges and what is its three-dimensional structure. So we believe that this streaming flow is a result of fluid structure interaction phenomena. The boundary of the egg cells are coated by thin hair-like micron-sized filaments called microtubules that are anchored to the cell boundary or the cell surface. These are some sort of highways or railroads for cellular transport. And just to give you a cartoon picture, you can think of these filaments as some sort of railroads as shown over here. And they are nanometer-sized machines consuming ATP and moving stuff along these highways. Okay? So on average, machines move from the base to the tip of the filament. And as they move up, they entrain fluid by carrying these heavy payloads. But by the virtue of Newton's third law, they also push down on the filaments. So these filaments are under compression. Now, if this compressive force is sufficiently large, these filaments can buckle. And as they deform, they generate additional flows. And then there is some sort of synergistic interaction between thousands of such filaments and the flows that they generate. And due to their collective interaction of deformation and the flows, there is a persistent emergent streaming flow that appears in the cytoplasm that you see over there. And if you look at the filaments on the boundary, they're all sort of lying down in one direction like seaweeds. So what I will tell you is about a theory that tries to explain the emergence of this flow, or to put this explanation in a mathematical framework, and tells you about how these things are organized in 3 dimensions. So in this theory, which I'm calling the active carpet model, I'm not interested in dynamics of individual filaments. Instead, I'm going to characterize the entire bed by a surface density C0 that tells me the number of filaments per unit area, and a surface polarity field N that tells me which direction these filaments are roughly pointing at one point. Okay? To make my life simple, I'm thinking of these filaments as rigid rods that are anchored to the cell surface, and at the anchoring point, there is a torsional spring that penalizes any deviation from the inward condition. So what that means is that they want to be really pointing radially inward. If you perturb them, that's penalized by this torsional spring. So this is really mimicking bending response of clamped filaments. So, and the action of these molecular machines that move along these filaments is caused into an uniform compressive force along the backbone end. So this is the structure of the problem. you have a thin microtubule layer. So this thin layer, size of this layer is much smaller compared to the system size. So this is really like a boundary layer problem. And in this boundary layer, you have stresses being generated. These stresses drives flows inside the cell, but these flows in turn reorients the dynamics of this layer, which in turn feeds back to the stresses. So there is a micro-macro coupling here, and that's what we want to model. So this is the evolution equation for the polarity field. First term is called Jeffreys equation. It just tells you how a rod reorients in presence of some shear flow. U is the flow which I want to compute. And then there is another piece coming from the restoring torque from the torsional spring that wants you to be pointing radially inward. To close the problem, I need to tell you what U is. And for that, I'm going to think about the cytoplasm as an incompressible Stokesian fluid. So we're in the low Reynolds number regime. It's highly viscous. Length scales are small, so the nonlinear terms don't show up in the problem. So this is really a linear constant coefficient homogeneous PDE, okay? And it has a no-slip boundary condition on the cell boundary. Now the action of this microtubule layer, the boundary layer, is cosine into a traction jump F bar across this interface formed by the tips of the microtubule vectors. This traction jump has two pieces. The first one is an active piece, which is very easy to understand, is that as these motors are moving, they're pushing on the fluid. They push along the direction N. How much do they push? Well, it depends on the force sigma of an individual motor, but there are many, so you just multiply them by the density. That's rho bar. The other piece is more subtle. It involves the rotational and interaction response of this whole microtubule bed. So as they rotate, they generate flows, and how they generate is encapsulated in the stress over here. So you can scale the problem. There are really two dimensionless number. One is a surface density, tells you number of microtubules per unit area, and then there is an activity number, which compares the forces from the motors to the forces coming from the bending in the problem. So higher this number, stronger is the active forces or active stresses. So if I want to summarize, this is the structure of the problem. You tell me what is the orientation of the bed. I know what is the surface traction jump. From that, I can solve the Stokes problem. And I've recast the PD as an integral equation over here, which is you can do because it's a linear homogeneous PD. I won't go into the details, but you can solve this equation for the unknown velocity U. And once you know U, you can evolve the polarity field N as follows. So before I show you some results, let me just give you some intuition about the problem, okay? So think about a half space where filaments are pointing radially or just vertically upwards. And when they're all pointing upwards, there is nothing interesting happening. There is no flow, so this is a fixed point of the problem. You perturb it, and whenever there is perturbation, what happens at the interface, you generate tangential stresses. These stresses generate a shear flow in the boundary layer, and the shear flow likes to rotate things. But now there is a competition between the springs that want you to be upright and the shear flow that's trying to rotate you. And when the shear flow is sufficiently strong, this whole bed deflects in one direction. And that's what's sort of encoded in this phase diagram of a stability analysis. If you increase density or if you increase the forcing, there is a critical value at which you have a supercritical pitchfork bifurcation, and we get to the streaming states where you spontaneously pump fluid. You can do this whole calculation inside a spherical egg, where when the filaments are pointing radially inwards, that's the fixed point of the problem. Then you perturb this state, and then you turn the crank, okay? So you expand these perturbations on the basis of vector spherical harmonics. It's a nice basis because these are eigenfunctions of our integral operators. They diagonalize everything. It's easy to compute the dispersion relation. I won't go into the details, but just want to highlight that this phase space looks very similar to the half space problem, and it depends very weakly on the radius of the confinement of the sphere. The L is equal to 1 mode is the most unstable mode, and this is how it looks. For the L is equal to 1 mode, you can see that this polarity field is sort of all lining up in one direction, and inside there is a large-scale vortical flow that's emerging. At the core, it's like a rigid body rotation, and in the boundary layer, there is a shear flow that comes from this eigenfunction. So that's all about linear theory, but you can do this problem in the nonlinear regime. You can simulate the equations, and that's what I'm doing over here. I've color-coded the surface of the sphere by an order parameter, which is close to 0 when things are pointing radially inward and close to 1 when things are aligned. If I let this system go, then immediately see formation of these aligned regions. You see the emergence of topological defects. That's a consequence of the Harry-Ball theorem. And then these defects interact, these aligned regions interact, they coalesce, and you end up with 2 + 1 defects sitting on the 2 poles of the spheres. You can look at the instantaneous flow fields. The -1 defects correspond to hyperbolic flows or hyperbolic points in the interior flow. And as these defects merge, and once you're left with these 2 + 1 defects, you see a large-scale vertical flow inside the cell. We can be actually quantitative about this process. So that's what I'm showing you over here. This is the steady state flow inside the sphere. It's strongly vertical and axis symmetric, but there is a swirl component that you see coming in this axis. And this is happening because there are filaments pointing radially inward along the two defects, and they're pushing fluid down. At the plane of the vortex, which is the plane transverse to the defect axis and passing through the center, the flow is entirely planar. And in this flow, the velocity increases roughly linearly from the core of the vortex, so like a rigid body rotation, but then quickly adapts to the boundary condition of the no slip. So you have a shear flow in the boundary layer. And I just want to highlight that this sort of computation not only qualitatively, but quantitatively agrees with the measurements that you get inside the cells. As you can see, we had a very similar profile, and the streaming speed is roughly 200 to 150 nanometers per second. So I should summarize. You probably realize that this is a high-dimensional system, but the dynamics that emerges is fairly low-dimensional. So you can go actually one step further and write down low-dimensional projections of these dynamics. That's one thing that we have done. But the real question here is that just like cows are not spheres, so are not eggs. So it's a natural question to ask what happens once we have large shape perturbations or actual shapes. And clearly, shapes plays an important role here. That's something that we're exploring. What's the bifurcation structure? Once you have small deformation, small perturbations, how transport and mixing happens, and what's the role of sort of perturbations and fluctuation? That remains unknown, but that's something we're looking at. So we have archive preprint on this stuff, so if you're interested, you can check it out, and I'm happy to take questions.
Thanks, Brato. Questions?
Thanks. I'm thinking about the beginning of your talk. You were sort of bringing up this aspect of transport, right?
Right.
And this kind of stirring that you're talking about. I'm just sort of wondering, is it possible, like, is the way it's used for transport, is it because you can control the stirring this way or that way, or is it just to mix things up enough so that things will meet?
Okay, so there are two pieces to the answer. The first one is that these are symmetry breaking bifurcations, so there is no chirality that emerges. So you do 50% time the experiments. In the experiments, you will find things going clockwise and counterclockwise roughly the same amount of time. That's one thing. The other thing is that the flows that we get also in experiments, and here they're not really good for mixing. These are really like rigid body rotations, right? So they are not efficient mixers. But what could be happening is that they're good for transport. So in a sense is that you can think of it this way, that it's actually known that once the egg cell fully develops and once it becomes a future embryo, it already has information of patterning, like where you need to form the head, where the wing, and so on. So there are localization of certain proteins that happen. And what could be happening is that so I'm not showing the whole picture, or maybe I can go back. So there is this whole sort of, so yeah. So as you can see, there are a bunch of cells here. So these are nurse cells that transport things, help the egg cells with things. And they transport proteins that get localized at different areas of the cells. So in absence of flow, these proteins would have diffused. That would have taken a long time. And maybe these flows are helping in sort of localization of these proteins, changing the time scale of the dynamics. That's our hunch, but that's something that we don't know yet. So it's probably interesting to look at things like first passage time problems with these flows.
So just from the experiments, do you see the vortices localize along some direction?
So you
with
mean,
respect to the
do you
cell
see vortices emerging, like the vortex core sitting around here? So yes and no. So what happens is that if you do a simulation inside a shape that looks like really this, so there is a very long transient where the vortex core actually sits somewhere like this, but eventually it transitions to a point somewhere at the poles. That's what we see, at least in our simulations and theory. And in the experiments as well, so this is, so there are different stages. This is, I think, a stage 10 experiment, but once you do the experiment in stage 11 and 12, those are more close to this ellipsoid, prolate ellipsoid shapes that I'm showing you. And in those cases, you see an axis symmetric flows and not like this one that I'm showing you over here. The nature of the flow changes with geometry.
And if you disrupt the flows, does it affect some of the patterning that you see at late stage?
So,
usage?
okay, so there is some theory for that, but I'm not completely convinced with them. I will tell you why, because the way you disrupt the flows is that you knock out kinesin, and then you say, oh, the flow is gone now, and now the patterning has been destroyed. But then kinesin is so important in so many other things. It's very hard to say that's exactly just disrupting the flow, and that's why it's stopping things. But that's typically how people do it, and there is evidence, strong evidence, if the flow doesn't happen, then the embryo is messed up.
Okay, so let's thanks Brato.
Okay.